Let's go ahead and start going through how we convert between RNA and DNA sequences. And before we start on that, I want to just go over some terminology that I'm going to be saying a lot during this video, so I want to make sure you guys know what I'm saying. So, the template strand, also known as the non-coding strand, is a strand of DNA that's going to make the mRNA. And your non-template strand, or your coding DNA strand, this is the DNA strand that does not make the mRNA. So, this can be confusing because you would hope that the words that start with N or start with non refer to the same thing, but unfortunately, they do not. So, on an exam, you could be given any four of these words, so it's important to know what all of them mean. And the way that I remember it personally is I remember template and non-template, because that's the one that makes the most sense for me, is saying the template makes the mRNA and the non-template doesn't make the mRNA. So I remember template and non-template, and then I remember the nons don't go together. So I know non-template therefore must be the coding strand, and template must therefore be the non-coding strand. And that's how I remember it. If you have another way you can remember that, that's fantastic. The next word I want you guys to be aware of is anti-parallel. That means one goes 5' to 3', the other goes 3' to 5'. And parallel would mean both going 5' to 3', or both going 3' to 5'. And then complementary base pairs. In RNA, this means adenine with uracil, cytosine with guanine. And DNA, it's adenine with thymine, and then cytosine with guanine again. Okay, so that's all the terminology that I want you guys to be aware of for this. So now we're going to talk about the relationship between the template strand, the non-template strand, and the RNA. And throughout this video, I'm going to be using the terms template and non-template because, like I said, those make more sense for me than the coding and non-coding. But keep in mind that on an exam, you could also be given coding or non-coding, so still know what those mean, even though I'm not going to be really using them throughout this video. So your template strand is anti-parallel and complementary to your non-template strand. This is something we knew already from chapter 10. So if we have our DNA, we know that they are anti-parallel and they are complementary. We also know that whenever we make the mRNA is made from 5' to 3', just like DNA. And that means that it's reading the template strand from 3' to 5', just like in DNA replication. So the template strand and your mRNA are also going to be anti-parallel, and they're also going to be complementary. So that means since both your mRNA and your non-template strand are anti-parallel to the template strand, so mRNA and non-template strand are both anti-parallel to the same thing, which means they both run in the opposite direction of the same thing. That means that they have to run in the same direction in order to both be running in the opposite direction of the same thing. And if they're running in the same direction, that means they're parallel. So your mRNA and your non-template are parallel to each other. In addition, both your mRNA and your non-template strand are complementary to the template strand. And if they're both complementary to the same exact thing, that means they have to have identical sequences. The only difference here is that since we're talking about DNA and RNA, they have that one nucleotide difference, where in DNA you have thymine and in RNA you have uracil, and everything else would be the same between them. So, in case me saying that out loud and reading this doesn't make total sense, I'm going to draw out this relationship. So I'm going to start with my template strand, and I'm going to label it 3' and 5', and I'm just going to do one example of every nucleotide, so A, T, C, G. Now I'm going to add on so this is my template strand right here. Okay. I'm going to go ahead and add on the other DNA strand, which in this case would be my non-template. And I know that my non-template is anti-parallel and complementary to my template strand, which means it must be 5' to 3' from left to right, and it's complementary. A pairs with T, T pairs with A, C pairs with G, G pairs with C. I'm going to rewrite this. And then finally, I'm going to draw my mRNA strand, which I know results from my template strand. So this is mRNA, and I know my mRNA is anti-parallel to the template strand. So my template is 3' to 5' left to right. My mRNA must be 5' to 3' left to right. And it's complementary. In mRNA, A pairs with U, T pairs with A, C pairs with G, G pairs with C. So now what I want you to do is ignore that green strand, ignore the template strand. Just compare your non-template and your mRNA. Notice they both run from 5' to 3'. They're parallel. From left to right, they're both going 5 to 3. And if you compare their nucleotides, the only difference is that T and U are switched, or in mRNA it's U and DNA it's T. So T or U, and then A, G, C. So your non-template and mRNA, parallel, same nucleotides, except T is U, or U is T. All right. So now we're going to go ahead and talk about a couple of common pitfalls, and then we're going to do some practice with this. So the first common pitfall that I see is people not paying attention to its being the template or non-template strand. So this could be two different ways, either in the exam it's written, below is a template strand of DNA, or below is a non-template strand of DNA, and people don't read the template or the non-template part and just assume it's one or the other, and then whenever they make mRNA, they'll get it wrong. Or the question will say, given this mRNA strand, solve for the non-template strand, but people don't realize that, and then they start solving for the template strand. So that's one. The next is not paying attention to direction. So if it's given to you 5' to 3', if it's non-template, that means it's parallel to mRNA. If it's template, it's anti-parallel to mRNA. So make sure you're looking at that, because there will be answer choices where the nucleotides are the same. The only difference is the direction. And finally, this is an important point. We're going to practice with this if this doesn't make a lot of sense right now. The answer choice might be given to you in the wrong direction, meaning that you need to read it backwards. I'm not going to spend a lot of time on that last point because I'm going to talk about it in the next slide, and I think it'll make more sense. So first, let's practice going from DNA to RNA. So let's say that I tell you I have a non-template DNA strand that is 3' GCC TGA 5'. And I want you to make RNA. So first thing that we need to notice, is it template or non-template? Okay, it's non-template. Is non-template anti-parallel or parallel to our mRNA? It's parallel. So if my non-template is written 3' to 5' left to right, so is my mRNA. So my mRNA would be 3' to 5' from left to right because they're parallel. I also know that my non-template and my mRNA are going to be the same nucleotides, except T is switched out for G. So that means it would be GCC UGA. So it's exactly the same. The only thing that I changed is T, I made it a U. And that might be an answer choice, or it might not be. The other answer choice that you might see that would be correct is just written the other direction. So your answer choice might be written from 5' to 3'. And all you have to do is look at what you wrote down and go the opposite direction. So 5' A, G, U, C, G, 3'. And I'm just going to write that down. So 5' A, G, U, C, G. Either of those could be or you'd only be given one of them on an exam. So you're not going to be given both on an exam, but you could be given one of those as the correct answer. So just because your non-template strand is written 3' to 5' doesn't mean that your answer choice would be. This is still completely correct, but it's written 5' to 3' left to right in an answer choice. So you have to make sure you're looking for both of them. Now let's do our template strand. I tell you the template strand is 3' TTC ATA 5'. So is this non-template or template? Template. Is the template anti-parallel or parallel to your mRNA? It's anti-parallel. So if my template is 3 to 5 from left to right, my mRNA would be 5 to 3 from left to right. So I'm going to write, 5' and 3'. And I also know that my template strand and my mRNA strand are going to be complementary to each other. So that means I want the complementary nucleotide. So I have TTC, the complement to that would be AAG. And for ATA, the complement to that would be UAU. All right, so you might be given the answer choice that looks like that on an exam, or you might be given it in the other direction, starting with 3' and ending with 5', in which case you just read the answer that you wrote backwards. So 3', UAU, GAA. And both of these are answers that are correct. Okay, let's look at our next one. This is just going the other direction, RNA to DNA. So if I tell you that your RNA strand is 3' UAG, CGA, 5', and I want you to find the template strand. So I'm looking for template. I know my template is anti-parallel to my mRNA. So if my mRNA from left to right is 3' to 5', that means that my template strand is 5' to 3' from left to right. And my template strand and mRNA are complementary. That means that I need to use complementary base pairs. So U is complementary to A to T, G to C, C to G, G to C, A to T. And that would be one option to write on your exam. Or your other one is just writing it the other direction. 3' T, C, G, C, T, A. And that could also be the answer choice. It would also be correct. So for our last one now, we have 5' GAU, UUA, 3' for our mRNA, and I want you to find the non-template strand. What do we know about non-template and mRNA? Well, we know they're parallel to each other. So if my mRNA is 5' to 3' from left to right, so is my non-template strand. I also know that my non-template strand has the same nucleotides as my mRNA, except U is T. That means it's going to be GAT, T, T, A. Or the other way this could be presented on an exam is just writing it in the other direction. 3' A, T, 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 A, G, 5'. And remember, you'd only be given one of the two on an exam as an answer choice, but you have to make sure that you're looking for both. So I would just recommend what we just did on the left side, where we said, okay, is it anti-parallel or parallel? And is it complementary or is it just switching T for U? And do that first without looking at answer choices. And then look for your answer choice as written. If it's not there, look for it backwards. And if it helps you to completely write it out backwards, do that. Because it really, depending on how long your DNA sequence is or your RNA sequence is, it could only take an extra 10 or 20 seconds. So if it helps you to just write out both of them in both possible ways and then look at the answer choices, definitely do that. But just keep in mind, it could be either way. And that is the end of that practice. So the next video will be going over initiation and elongation in prokaryotes.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.